Making your research on animals transparent. From the beginning to the end.
Why we need transparency.
Scientific research has always been based on openness as science is built on science. Information as well as methods, ideas, and data are shared within the community of researchers. Yet, for long, research has been visible to the big public mainly in the format of scientific breakthroughs.
Research results are first published in scientific journals aimed for, and read mainly by, peer scientists.
Eventually, very few of these results are filtered to big public and can be found on the pages of local newspapers.
However, the transparency of research projects as a whole, not only the selected end results, has become a major issue in biomedical research that involves work on animals.
With the increased awareness and concern over experimental work involving animals, researchers are requested to promote openness towards laymen as well.
Hence, it is essential that authorities give clear instructions on what to report, and that there are open platforms from which this information can easily be found. What is an NTS?
The first step in all research on animals is applying a project license that authorizes the work. Since 2013, the license application within the EU has included a non-technical project summary NTS. NTS is specifically aimed at non-specialist audience. It explains project design, including objectives, harms for animals, and how the 3 Rs are applied. It is important that NTS clearly describes how harms to animals are minimized, and benefits maximized in your project. Publication of NTS fulfills the transparency goal of the EU directive and is obligatory in EU countries. Starting from 2021, Finnish NTSs are published by the EU. The previous ones are available at the Regional State Administrative Agency. How to compile a good NTS? Make it understandable and clear.
It is not a new thing for researchers to know their audience. Researches are used to adjust the content and language used when presenting work to peer scientists, or
to students from their scientific field, or
when they talk about their work with their family and friends. NTS should be aimed at the last mentioned group of people. The goal of transparency is only met if this audience understands the content of NTS. Hence, sentences should be kept short and expressions easy to understand. Technical terminology and laboratory jargon should be avoided.
This said, it should be clear that the text in NTS differs greatly from that used in the rest of the project proposal, which is aimed at experts in the Project Authorization Board.
What is the content of NTS? Consider NTS as a standalone summary of your project. It should clarify why animals are needed in your study and what kind of benefits are expected to result from the project. NTS includes information on which animal species and how many animals are used and what are the harms to the animals. A very important part of the NTS describes how the 3 Rs are applied. Despite this detailed information, NTS is anonymous, so do not include names or addresses.
Let's see how researchers who study how different diets increase the risk of particular liver disease have compiled the NTS for their new and exciting project.
Our study is called The Effects of Dietary Sugars on Liver Function. A drop-down list of different purpose codes is inbuilt in the new NTS form. Our research falls into the category of translational and applied research, human endocrine, metabolism disorders.
The objective of our study is to increase the understanding of the generation and progression of the non-alcoholic fatty liver disease, NAFLD. A significant potential long-term benefit is the generation of new dietary recommendations that aim to reduce liver diseases and thereby improve life quality.
Studies suggest that a high intake of fructose may increase the risk of liver disease in which too much fat is stored in liver cells. This kind of fatty liver disease can also lead to liver inflammation and further liver damage. Since fructose is increasingly common in the Western diet, it is important to understand what kind of metabolic effects its excessive use may cause.
Predicted harms. The experimental groups are given fructose supplemented drinking water. This causes a significant increase in body weight, blood plasma triglyceride and glucose levels. Notably, these changes do not affect animals' subjective welfare. The study lasts for 6 months and we will follow the progression of the disease using a non-invasive imaging technique. Imaging of anesthetized animals is done at 2, 4 and 6 months. We also collect blood samples once a month. Both sampling methods cause transitory mild distress and pain to mice. At the end of the project, animals are humanely killed, and tissue samples are collected for analysis.
Application of 3 Rs. Replacement. Other alternatives have been considered, including in vitro models, organoids, and the use of a collection of human postmortem samples. In our previous study, we examined cellular responses to metabolic challenges using cell cultures. However, the core aim of this project is to study changes in different metabolic pathways and their effects at multi-organ level. This is not possible in the above-mentioned alternatives but requires observation of the physiological responses of a living organism.
Reduction. With the help of a statistician, we have calculated animal numbers to ensure that the maximum amount of data is obtained from each animal, and that scientifically robust data can still be generated. To confirm the feasibility of our study design, we will perform a pilot study on a small number of animals. Once the project is completed, animals are killed and tissue samples are collected for analysis. Sharing the material with members of our research group will further reduce animal numbers. Sharing will also increase the information gathered from a single animal.
Refinement. Harm from handling and sampling is minimized by training the mice so that they are easy to handle. This way, imaging and blood sampling do not cause major distress to them. Over the duration of the project, animals are frequently monitored and scored using project-specific welfare score sheets. This ensures that no animal exceeds a strictly predetermined level of distress. Humane endpoints are implemented immediately if animals experience unexpected adverse effects.
Expected harms. The harm and distress caused to the animals is at most mild. I notice that the new NTS sheet asks how many animals are used in each severity category. Non-recovery, mild, moderate, and severe.
That's it. Our project from the beginning to the end. I didn't think that we could crystallize it so efficiently and clearly. I am sure that our NTS will enhance openness and transparency around animals in research, facilitate understanding of different animal use areas amongst the public, encourage scientists to develop and improve their communication skills, and better explain their research interests to the public.
Improve the quality of the scientific information available to the public and avoid the spread of misinformation. Support the sharing of good practices in relation to the 3 Rs. Support evidence-based policy making by competent authorities. What do you think?